passed out a sheet to you all that goes with this lesson that we'll of course work on once we're finished going through the notes. And it is asking you to draw, and I just want to reiterate before anyone freaks out that your artistic prowess is not the thing that I'm marking, right? You can all draw circles and squares, I bet, and lines. So I'm not looking for works of art, but rather, can I tell what you're representing? And all of the slides of the notes have samples of what it could look like. What I do want to make mention of is I do not want you to just take a picture from the internet and put it on your assignment. That's not the goal. Now we're going to focus today on a synapse. We've heard the word used before. We've also heard a word that sounds like this, synapsis. Synapsis was the word we used to describe pairs of homologous chromosomes lining up and forming a tetrad during prophase of meiosis. And I just want to point out, right, it's not the same thing, right? That was to do with chromosomes lining up. This, the synapse, this is the connection between two neurons or between a neuron and an effector. So much like yesterday where I had one picture but several slides, that's kind of what we're looking at today. So I'm going to talk about the things that you see on this picture and some information that goes along with it. So a synapse is, like I said, the connection between two neurons. The word synapse means link or junction point, and especially in the context of your neurons connecting to your muscles, which are effectors, we use the term neuromuscular junction. That's terminology that you would hear. And if you're going to study anything to do with this kind of stuff moving forward, that's what you'll hear, is neuromuscular junction. That's the name for a synapse between a motor neuron and a muscle cell. It's still just a synapse, right? But it has its own special name. So especially if you're looking stuff up or when we're working on the assignment, if the drug that you choose to look up affects the neuromuscular junction, you're like, what the heck, man? It's just the synapse between a neuron, a motor neuron, and a muscle cell. Now, the thing that I need you to understand here is that a synapse involves physical space. One neuron is not physically touching the next one. There is a gap or a space between them. So this space between those two neurons is called the synaptic cleft. And so something is going to have to travel from one neuron to the next through the synaptic cleft, and that's what we're going to talk about. So the influx, when we're talking about ion movement, or we could just say the impulse, will arrive at the presynaptic terminal. Now, if underlining things with a smartboard pen would work the way I thought it would, I would underline the word presynaptic. When you have a word that starts with pre, it means it comes before something else. The presynaptic axon terminal is the structure that looks like this, like an elephant's foot. It'll have that kind of round shape at the end of the axon terminal. And so if I were to imagine zooming out on this, this would be the axon up here, extending up towards the cell body and the dendrites of that neuron. So we're focused in on the end of the axon and the axon terminal. So you would need to be able to identify where the presynaptic neuron is. And that's this part, the part that comes before the synapse. Now, how do I know that's before and the other part is after? Well, I'm going to see some visual cues here that are going to help me out. The first thing that I need to recognize is that information, if you recall, flows in one direction, from dendrite to the soma, through the axon, all the way down to the axon terminals. So the axon terminal is the end, we'll say, of one neuron. The dendrites, we can see labeled here, that's the beginning of the signal transmission in the next neuron. In the axon terminal, like you see here, the arrival of the influx or the impulse causes the opening of some channels, specifically calcium channels. And then there are chemical messengers called neurotransmitters. Neurotransmitters, you can see over here, it's pointing to the little black dots. That's usually how we draw a neurotransmitter, some sort of tiny shape. Sometimes there'll be dots, sometimes there'll be like little triangles or little squares, but some sort of little generic shape is how we represent a neurotransmitter. They're actually fairly complex chemical structures, but this isn't chemistry. So we're just going to represent them with a tiny little geometric shape. I can see that these neurotransmitters are in the synaptic cleft, but I can also see all of these circles up here are full of those little black dots. That's a clue telling me, aha, that's where the neurotransmitters are coming from. So I know that's the presynaptic neuron. But these chemical messengers, the neurotransmitters, are going to carry the signal from one neuron to the other, or if it's an effector, from the neuron to the effector. Now, so far, does anyone want to ask anything about what's been written there? I haven't talked about everything on the diagram because the same diagram's on the next page, and we'll keep going. One of the things people sometimes ask me is, how many neurotransmitters are there? Lots is the answer. We're not going to talk about all of them. That would take too long. We're going to focus on a few specific ones, and you'll see in a couple of slides. The main one we'll look at is acetylcholine, but there are other neurotransmitters that you've probably heard of, dopamine, maybe serotonin. Those are sometimes things that people are like, oh yeah, I've heard that word before. They're all examples of little chemical molecules that can cross the synaptic cleft. So perhaps your question is, okay, so nerve impulse comes, calcium channels open, this little molecule is going to cross the synapse. What does that even do? How does that even work? How would that little chemical molecule tell the next neuron that there was an electrical impulse causing action potentials in the previous one? So same picture, I'll just move this out of the way. Whoops. Okay, same picture, but now we got to understand how the neurotransmitter is getting from one side to the next. So up here talks about synaptic vesicles. Those are the little circles that you see in the presynaptic neuron. There are a whole bunch of them. You hopefully remember hearing about structures like vesicles or vacuoles in Science 10 when you talked about the parts of the cell. It's a little sac surrounded by a membrane. It holds stuff. In this case, it's holding neurotransmitters. These little vesicles move towards the edge of the axon, towards the membrane. And because of the calcium that was stimulated previously, this is stimulating exocytosis. So perhaps you remember exocytosis. It's another Science 10 idea. If you look right at the edge of the membrane of this synapse, you see exocytosis happening. It's where one of those little sacs that has a membrane approaches the cell membrane. They are both made out of membrane, so when they touch each other, they fuse and open up. So right here on the left, and kind of in the middle a bit and on the right, you can see 3 synaptic vesicles that have started to fuse with the cell membrane. The result is that they open up and they spill their contents out into the synaptic cleft. Now, if you're thinking how long does all of this take, a millisecond or maybe less for the neurotransmitters to get from the presynaptic neuron to the post means after, postsynaptic neuron or effector membrane. So this movement, a millisecond or less, right? It's a really quick thing that's happening. Once the neurotransmitter has been released through exocytosis and has crossed the synaptic cleft, it will bind to some sort of receptor. So here, the receptor is shown as just like a little divot. On some of the pictures that you'll see on practice questions, it's shown as a structure that sticks out of the membrane a little bit. It's a spot for the neurotransmitter to go. It's usually represented with the shape that matches the neurotransmitter. So if we drew the neurotransmitter as a triangle, it'd be like a triangle shape. Or if we drew the neurotransmitter as a circle, there'll be a little circle shape. That's how the diploma tends to like to do things, matching little geometric shapes. When they bind to these receptor proteins that are on the membrane of the dendrite of my postsynaptic neuron, several things can happen. So, first possibility. If the neurotransmitter is what we call excitatory, they will cause sodium channels to open. What happens when sodium channels open? Depolarization. You get an action potential. If the neurotransmitter is inhibitory, the receptor will cause potassium channels to open, meaning hyperpolarization will happen. Kind of what happens at the second to last portion of an action potential. Now, I've simplified here. There are actually other ion channels that could open. The key is excitatory neurotransmitters depolarize the membrane. Inhibitory neurotransmitters hyperpolarize the membrane, make it harder for it to have an action potential. If the threshold is reached, if enough depolarization overall happens, then action potentials can be initiated, just like we talked about before. And then, like we mentioned a couple days ago, the cell body, or the soma, its job is to integrate all of the information it gets from all of the dendrites. So from all of the receptors on all of the dendrites receiving neurotransmitters from different places, it'll integrate those signals and determine the combined effect. So maybe one side of the neuron is getting inhibitory neurotransmitters, and the other side is getting excitatory, and they'll cancel each other out, and there'll be no action potential. Maybe there's more excitatory neurotransmitters than inhibitory, and you will get an action potential generated. Okay? So these are possibilities. It's not necessarily going to happen in every scenario. Those are things that could happen. The other thing that people will want to ask here is, which neurotransmitters are excitatory? Good question. Sometimes neurotransmitters can be excitatory in one context and inhibitory in another. So I don't want to associate one necessarily with those terms. What you do just need to know is excitatory neurotransmitters cause depolarization, start action potentials. Inhibitory ones cause hyperpolarization, limit action potentials. Now I'm just going to pause here, see if anyone wants to ask anything. Maybe you have questions about the picture, maybe you have questions about the words. So we're going to take a look on the next slide at a specific example of a neurotransmitter. I have on purpose chosen a different picture for the next slide because I want you to get in the habit of recognizing the key elements of diagrams rather than hoping it's the exact diagram that came from the notes, which is pretty unlikely overall. So, different picture, but I hope you recognize there's lots of similarities there. We're taking a look at acetylcholine, which is very commonly abbreviated ACH, because acetylcholine is a really long word, and scientists, I like to say, are lazy and don't want to write long words. That's why we use symbols for everything. So acetylcholine is a neurotransmitter that crosses a neuromuscular junction. So specifically, it's coming from a motor neuron, and it's going to bind to a receptor on the membrane of a motor or of a muscle cell. Now, if I take a look at the picture up here, I see a lot of the same things I did on the previous one. Here's the presynaptic axon terminal. How do I know it's the presynaptic one? Well, it's got these little vesicles that have neurotransmitters inside of them. That's my clue. I can even see two spots where exocytosis is happening. Here's my postsynaptic structure. Because this is a neuromuscular junction, that would be muscle cell right there. If it was a neuron connected to another neuron, that would be the dendrite. But this is the postsynaptic side. So this space here is the synaptic cleft. Here you see receptors drawn, like I mentioned before, shape that matches shape. Since in this picture, acetylcholine is represented as little sort of circular things, the receptor has like a little circular spot for acetylcholine to go. Now, perhaps you're saying to yourself, okay, this is great, but how does this know when to stop? I've let a bunch of acetylcholine out into the synapse. What happens next? it just sits there forever and keeps binding to the receptors, that would be a really ineffective thing. And so your body has ways to prevent this. For a lot of neurotransmitters, the way we prevent continual excitation or continual inhibition is to use an enzyme. Now, in the case of acetylcholine, the name of the enzyme is cholinesterase. And so if I look at that word, I say, oh, it ends with ASE. That's how enzyme names tend to end. They don't have to, but if I see a word that ends that way, I think really hard and I think this is probably an enzyme. So cholinesterase is the enzyme that's going to work to break down acetylcholine. So perhaps you see this red triangle. I know the color difference isn't super distinct because there's purple in it too, but it's labeled acetylcholinesterase. That's our enzyme, and its job is to break down the neurotransmitter. Essentially what it does is it breaks it into two pieces, thus rendering it ineffective. Because it's breaking it down, that causes my postsynaptic channels to close, so I end the effect. And there are lots of drugs whose function is to affect these enzymes. In particular, my example here is about sarin. It's a neurotoxic gas. It's used in, among other things, some insecticides. So this is how it works. It destroys the function of the enzyme, so it inhibits the enzyme. That means the enzyme cannot break down the neurotransmitter, and the neurotransmitter just stays in the synapse, keeps binding to the postsynaptic receptor, and in the case of the neuromuscular junction, results in constant muscle contraction, which results in death in this scenario. There are lots of other drugs that work on this premise. You'll see in the assignment we're going to look at today, we're going to look at the recreational drug category, but there are also drugs that are used because doctors have prescribed them. Lots of antidepressant type drugs work in this pattern by inhibiting the removal of a neurotransmitter from the synapse so the effect can be continued. And so we're going to take a deeper look at that type of thing. Now, does anyone want to ask anything, especially about the way a synapse looks and what the parts are? Because that's what you're going to have to put on your assignment. Okay. So then my last thing, and will be the end of the notes, is just to let you know, like I mentioned before, there are other neurotransmitters. Here are a few of the ones that get mentioned frequently. So it's the ones that I want you to be aware of. Dopamine was one of them. Dopamine is a big player in terms of movement control and pleasure, feeling happy, feeling good. Dopamine is researched in a bunch of different contexts because people who have motor control issues, for example, people who suffer from Parkinson's, dopamine is a big player in that scenario, but it also makes you feel happy and good about things. So it has a lot of structures that it affects. Serotonin is really important for sensory and temperature perception. Endorphins are your natural pain relievers, so your body naturally makes those. Norepinephrine, I'm mentioning, and we're going to come back to that one. There's a category of molecules, norepinephrine, epinephrine, or adrenaline, noradrenaline. They get lumped together even though they're not chemically the same molecule, but they have similar functions. This is an example of a neurotransmitter that's involved in your response to stress. But sometimes things are called neurotransmitters, and other times they can be called hormones. It kind of depends where they're being produced and where they are traveling to. So we'll come back to this one again because we're going to talk about stress again in a little bit. But these are just some examples of neurotransmitters that you have. Now that's the end of notes and note-related stuff. What we do need is to take a look at your assignment. So if that sheet is behind things, maybe pull it out to the forefront. So it tells you to refer to this page, so I'm just going to have it open here. Really just for the content. I don't want you to answer the questions that they have there. I want you to answer my questions. But this page, 383, so it's the one right before we were looking at in the digital book, has some examples of different types of drugs and how they affect the nervous system. Now, it also gives some description of how it affects the body as well. We're going to focus just on the nervous system part. Specifically, how are these drugs impacting the way the synapse works? That's the focus of what we're doing. So there are several drugs listed here: nicotine, marijuana, ecstasy or MDMA, cocaine, and methamphetamine. You can choose one of those because here it is, the content is there, but you don't have to. If you would prefer to, and I always have to choose my words carefully, if you would prefer to choose to research a different drug, not to choose a different drug or do a different drug, which sometimes I say, but if you want to do your assignment about a different drug, you are welcome to. Just maybe check with me what the drug is. Some drugs are easier than others. Sometimes people think it's one drug, but it's actually a whole category of drugs. So what I'm saying is, pick one of these or pick a different one, your call. But the assignment is the same either way. You're going to draw two labeled diagrams, and it's really important that they're labeled. If I don't know what I'm looking at and you don't know what you're looking at, then it's not super helpful. I'm not looking for artistic prowess, I'm looking for, can I see the structures? The first one is a synapse functioning normally. I would be looking for, where's the presynaptic neuron? Where's the postsynaptic neuron? Where's the synaptic cleft? Where are the vesicles with neurotransmitters? Where's the receptor? Those are the types of things that I just showed you about in a typical synapse. Then, you are going to draw a synapse again, but after the drug that you chose has been introduced. Really focusing on what's different. Has the drug made there be more of your neurotransmitter in the synapse? Has your drug affected the enzyme that's supposed to break down your neurotransmitter? Has your drug acted as a competitive inhibitor and blocked the receptor for your neurotransmitter? So I need to see very clearly, and I would suggest that you write the word difference and point an arrow at it so that it's super obvious what the difference is. But highlight the differences. Make sure that it's clear what was going on before, regular synapse, and now that this drug has been introduced, what was different. Now, different drugs affect different neurotransmitters. So, if you take a look up here, for example, this one mentions dopamine. This one talks about, this one talks about serotonin. So, please make sure that you label your neurotransmitter accordingly. If your drug is affecting dopamine, label your neurotransmitter dopamine. If your drug is affecting acetylcholine, label your neurotransmitter acetylcholine. Okay? Now, does anyone have questions they want to